Thanks, Soil CRC, for this opportunity for us PhD students to come here and present our research. My name is Henry Luutu. Good afternoon, everybody. Today's presentation, I'll present on biowaste to value, but I'll keep asking myself, do plants and soil organisms really agree to this? So this is the presentation. This is the presentation I'm going to dwell on. So basically, my presentation has been my PhD, basically my PhD has been on recycling of waste into soil as soil amendments, but my focus has been on hydrothermal carbonization. But then, what is hydrothermal carbonization? So basically, hydrothermal carbonization is a thermochemical treatment approach that converts waste into carbonized material. So when these carbonized materials are produced, they can be used as soil amendments. So the process is simple. So biowaste or any other material can be added into the reactor, and then it can be boiled or cooked at a given temperature, and then we kind of get two products. So one product is called hydrochar, which is more or less like biochar, but because it's produced in a different kind of system where we add in water, so we call it hydrochar. Hydro basically means water. And then we have also another byproduct, which is a bio-oil that can be used for diesel engine. But for us, our focus in this research has been to use it as a soil amendments. Biochar improves soil physical properties, soil properties, generally all the nutrient component of the soil. But some researchers have shown that biochar can also have some bit of negative impacts on the plants because they assume that it has some toxins that come along when it's being produced. So in this study, I focused on trying to assess what exactly happens if hydrochar is applied in the soil. So I tried to do some plant studies, and one of the plant studies, I did it under limited nutrient to see what exactly happened if hydrochar is applied in the soil under low nutrient. So these are the results. Because of time, I'll only dwell on the biosolids. These other materials, we use them, the chicken manure, digestate, punch, pig manure, rice straw, and sawdust. We use all these materials to produce hydrochar, but because of time, I'll only focus on the results from the biosolid hydrochar, because hydrochar is a treatment mainly appropriate for wet material. So we have a lot of waste streams that have high content of moisture, so hydrochar can be a very good alternative for pyrolysis. So that's why I will focus on biosolids. But when you look at these results, you realize that addition of hydrochar in the soil reduces plant material, rather plant biomass. So we can look at the results under 200. Where's the pointer? Oh yeah, I've got it. So when you look at the 200, it reduced plant biomass, and also when you look at 260, it also reduced plant biomass. When I talk about 200 and 260, I basically mean the temperature at which we produce this material at. So they all reduce plant material, but then we could not really tease out what exactly caused this reduction in plant material. So we could think either it's a reduction in nutrients within the soil due to mineral nutrient immobilization, or it can be toxic, as other people have always been associating it to. So we were thinking of what exactly to do. So we had to run another study where we add sufficient nutrients to the soil so that even if there is microbial immobilization where the microbes come and take up the nutrients and they reduce the nutrients for plant uptake. So we try to add in more nutrients so that the plant can have sufficient nutrients. Even if the microbes take up the nutrients, the plants will remain with sufficient nutrients for growth. So we tried this in experiment 2 where we called it luxury nutrient, and then we just realized that the biomass boomed. So we can see it here that this was initially low, but it's up here now. So it means that it has crossed over to the control. So it was above the control. But of course, now with the 260, the biomass really remained low. So basically, this one means that microbial actions that cause immobilization were playing a part in this. So this was rectified in this in this experiment. So at 200 degrees of hydrothermal, you find that those ones were not toxic. So we proceeded because we realized that hydrothermal can either be toxic if you produce them at higher temperatures, but they are not toxic, just as Ore's reports put it up, that every hydrothermal might be toxic. So we tried to test them, the earthworms, to see, because earthworms really represent the community of soil faunas in the soil. And then we realized that earthworms for the biosolids, still, as I told you, that I'll dwell on the biosolids, sorry. Yes. Yes, so when we tested earthworms, earthworm is one of the tests that is used to test toxicity or pollution in the soil. Usually, if the soils are toxic, the earthworms will basically run away from those soils. So we use this test to test our hydrothermal, and we just realized that the hydrothermal were not toxic to the earthworms. The hydrothermal made from biosolids. So this line above here basically means that this habitat is not safe for soil faunas or earthworms. But of course, down here, it means this habitat is safe for these organisms. So the 260 that also had an issue with the plant was still toxic here, and then the 200 was safe for the plant. So basically, we can conclude that hydrothermals produced at higher temperatures could be toxic, but of course, not every hydrothermal that is produced is toxic. So basically, that was my study. But of course, I did some other studies, including enzyme activities, PLFAs, and yeah, but because of time, I can't present everything. Yeah, thank you.